go back, please. What you should have seen here is that with the time of incubation with HNSCC and CpG, CpG would completely reverse this. So IL-10 would be lowered, IL-10 would be lowered, IL-1 would be lowered, and interleukin-6 would come up massively. So CpG would completely reverse this cytokine shift induced by HNSCC. So it would be a very worthwhile treatment target. Next. So now we have migration, we have function in terms of cytokine production. Now we have the most important thing of DCs. They have to present the antigen to elucidate a complete T-cell response. And we do have an assay where we incubated MDCs with HNSCC, let them then pick up antigen and present it to T cells. And T cells should then lyse the target and by this present their capacity. And what you see is that the longer that MDC are incubated and supernatant of head and neck cancer, the less antigen can be presented and the less efficient the T-cell response works. So taken together, there is the clear model of dysregulation of myeloid dendritic cells by head and neck cancer, because the head and neck cancer accelerates the migration of immunologically defect, tolerance-inducing myeloid dendritic cells to the local regional lymph node. It actually hinders the right cytokine production, it accelerates the wrong cytokine-producing cell, and it hinders the presentation of antigens, and by this activation of T cells. As I've shown you, or actually could not show you, CpG would be the right drug to stop migration, to stop IL-10, to lower IL-1 and induce IL-6, and by this revert the whole damage HNSCC would cause in the dendritic cell system. Next. Let's come to the other cell, the PDC, the cytokine secretion one. This cell sits in HNSCC. We've been the first to describe that. And in the majority, this cell is not activated. So they don't show CD86, they don't show whatever marker you'd like to see. They're just plain dumb DCs. Or actually not, because they are forced into a Th2 milieu by the tumor, which is tolerant-inducing. They're reduced active, and they have basically no to very low secretion of interferon alpha. So they're completely paralyzed into a Th2 milieu by the tumor. Next. How come? How is it that interferon alpha is not secreted anymore? And now we've done a real-time PCR of all the TLRs, but we also have the protein levels in the meantime, which are one-to-one basically. And what we see is that the TLRs that would be necessary to use interferon alpha, predominantly TLR9, but also TLR7, are taken down from the surface of the cell. So the tumor makes a TLR downregulation on PDC. So the PDC, even if it would have the right stimulus, would not be sufficiently able to react. And this causes this Th2 situation. Upon CpG, if you put CpG into the supernatant, we can upregulate the TLRs, not completely, but we can halfway restore this downregulation. Next. The other thing is, it's not just a question of TLR regulation, but it's also a question of activation. So if we take single-cell suspensions, so this is a flow cytometric analysis, and we stain CD86 as a marker for activation and the characteristic for PDCs, we can clearly show that tumor itself shows no activation, medium would show a bit of activation, medium plus CpG would cause a major activation of formerly tumor infiltrating PDCs. Next. Same holds true for the tumor-draining lymph nodes. So PDC and MDCs would, in the tumor-draining lymph node, would completely be recovered if they could see the equivalent CpG they would need. And the activation would be much higher than in tumor or in medium. The other thing is that if I get the DC activated, the question is what happens to the whole network the cell is acting in, one of which is the CD4 and CD8 T cells that are infiltrating the tumor. And what we see is that CD69, the activation marker of these cells, is very low in the tumor baseline. With media, it's okay, but if you put CpG to it, the T cells would be activated indirectly by PDCs and the interferon alpha that PDCs generate. That also holds true for the lymph node. You see lymph node, tumor burden lymph node, no activation, low activation, and high activation of CD8 T cells. So indirectly, we could restore the whole immune system by CpG. Seen as a total model, we come to this point. We do have a tumor cell that causes immature, resting Th2 secreting PDCs. We have a dysregulated MDC. We have the situation of tolerance, and by producing interleukin-1, tumor growth promoting situation. Next. Result is the whole network, not only T cells, but also NK cells, are completely anergic and do not eliminate the tumor. Next. Upon CpG, next, or any other type of pump, predominantly what we currently look at, immune stimulatory RNA, the DCs would be activated, next, elucidate with their cytokines or directly in contact the whole immune response, which then would generate, next, T cells that could very efficiently, next, eliminate the tumor cell, and by killing the tumor cell, the substances needed to re-stimulate, this is kind of autocrine cycle, the next generation of disease would be produced. So we need some type of this drug in whatever delivery system to then generate a sufficient immune response. Next. I want to thank all my co-workers that helped me build up this network. Thank you very much.